Thank you, Mohammed, for this nice presentation, and thanks to all the speakers for their insightful presentation. So today it's my honor to present our latest data within the International Conference and Workshop on specialized cell culture. Our project is entitled CRISPR-Cas9 editing of the ATM gene in mammalian cells. It was elaborating during my postdoc at the Unit of Cellular Networks and Molecular Therapeutic Targets in Regina Elena National Cancer Institute, Rome, Italy. So ATM, or ataxia telangiectasia mutated gene, is a threonine kinase. It's a key sensor of DNA double-strand breaks repair, and it contributes mainly to a DNA damage response, to the regulation of cell cycle checkpoints, survival, and DNA repair machinery through the phosphorylation of several targets such as P53, CHEK2, and BRCA1. Sorry. Okay. Okay. So at the beginning, it has been discovered as a gene responsible for the AT disease, which is an autosomal recessive inherited disorder characterized by progressive cerebral ataxia, immunodeficiency, radiosensitivity, and very high cancer predisposition to very numerous cancers. And it is now considered as an intermediate risk breast cancer susceptibility gene. So advances in high throughput technologies of sequencing have generated detailed catalogs of genetic variation in both cancer patients and in the general population. Nevertheless, our ability to predict the phenotypic consequences of each genetic variant remains poor, limiting the medical impact of sequence information. This problem is mainly highlighted by the large number of variants of uncertain significance, or VUS, which are identified in both the driver and actionable genes, and also the still unmet demand of functional characterization of these variants. So within this context, the most reported variations so far in the ATM gene are the ATM nonsense truncated mutations, resulting in ATM protein disruption. And in the homozygosity state, they are responsible for the AT disease. Less well characterized but more frequently are the ATM missense variants, which do not directly disrupt the protein. They induce less effects on ATM function and stability, and their contribution to the disease is frequently unclear. More importantly is the fact that 50% of these ATM missense variants reported so far are variants of uncertain significance. So the main purpose of our project is to generate clones of human non-transformed mammalian cells with a collection of ATM variants in order to study the phenotypes and their consequent tumorogenetic potential. So to be done, we have opted to utilize the revolutionary CRISPR-Cas9 genome editing in order to generate knock-in clones with 12 single ATM variants. These variants are 10 missense variants, 2 nonsense variants to be used as negative controls. And among the missense variants, 5 are currently classified as VUS. So briefly, the revolutionary CRISPR-Cas9 system is a phenomenal gene editing tool referred as genetic scissors. It allows modifying genes through double-strand breaks, which are repaired through 2 different pathways, either by non-homologous unjoining or by homology direct repair. So within the system, the single guide RNA, it will guide a Cas9 nuclease in order to cleave the DNA in a specific genomic locus based on sequence match, resulting in double-stranded DNA breaks, precisely 3 nucleotides of an NGG protospacer adjacent motif referred as the PAM. So within this respect, we performed an in silico analysis to identify 12 variant-specific guide RNAs and 12 relative 180 base pair long single-strand oligodeoxynucleotides in order to be used as repair donor templates for homologous recombination. So each single-strand ODN is specifically designed with a single nucleotide substitution corresponding to the variant of interest, and one or two other single silent substitutions for preventing the Cas9 nuclease recutting. So then we co-transfected RPE1 and ELA cells, which were our models for transfection optimization, with each variant corresponding Cas9, guide RNA, and puromycin resistant genes plasmid, together with matched single-strand ODN. And after puromycin selection, the resistant cells, they underwent single cell plating for clone formation. Cell pellets were then collected from all the clones of each variant for genomic editing verification. So indeed, our strategy for no-kill clones screening was, we saw it really accurate and correct to follow the strategy of extracting genomic DNA and or performing afterward rounds of PCR, restriction analysis, and or Western blot. So these techniques, they were applied in the purpose of qualitative screening of the presence or the absence of the variant of interest, and finally sequencing for the precise screening and confirmation of the potential edited sequence. So to be done and to assess this methodology, we first try to generate no-kin clones with 3 variants. The first one is a missense variant, clinically classified as benign, likely benign. The second was a missense variant classified as VUS. And the third variant was our negative control, and it was a nonsense variant classified as pathogenic. For the in silico analysis for the restriction screening, we have used the web tool CRISPR Cruncher in order to seek for enzymes having one or more specific consensus sites, including our variants of interest locus or adjacent to them, in either in the PAM sequence or in the silent mutations in the guide RNA, in the purpose of showing variations between the wild type sequence and the edited ones. In addition, we have made use of the NAPCutter tool in order to have further comprehensive view of the theoretical digest sizes produced by the selected restriction enzymes for our variants of interest. In the case of the nonsense variant, we could not identify a consensus site, but since it causes the formation of premature stop codon leading to the production of a truncated protein, so the presence of our protein of interest was then checked by Western blot. So by following our strategy, the screening of all the multiple obtained clones was performed by rounds of PCR and restriction analysis, and using the selected restriction enzymes. Nevertheless, all the screened clones resulted to be wild type, meaning non-edited. So from this point, we resolved to improve the efficiency of our system. Thus, we adopted a different strategy. We first changed the cell model by using cells of the human mammalian non-transformed MCF10 cell line in order to increase the probability of having no-kin clones by transient puromycin selection. And our second strategy was to redesign the single-strand ODNs in order to, and this redesigned as ODN were shorter than the first one, and thus in order to favor the homologous recombination. So by restriction analysis, as you see, we have found potential cell clones carrying the variant of interest, finally. So we were happy. So we Sanger sequenced those clones and we came up with 2 Nokin clones. And as you see in this particular case, so 64% of, we have found 64% of the homologous recombination, 60% of the wild type, 3% of a -3 deletion, and 1% of the -4 deletion at passage 2. So as we were happy, so we then repeated this sequencing after few cell passages in order to further check the clones. But to our surprise, Sanger sequencing of the isolated cell clones revealed that the variant of interest in the potential Nokin clones, they were lost after few cell passages in favor of the wild type sequence. And since it was, as you see, not possible to keep and obtain single cell clone population, so we decided to move on by analyzing mixed populations, investigating the 3 variants, including the VUS, the pathogenic, and the benign variants, in order to identify this time the variant-specific cell behavior. So we opted to perform NGS of transfected cell mixed population DNA at 3 different consecutive points, which are 6, 13, and 20 days. So as a result of the NGS sequencing, we have seen that the collection of the cells at the 3 different time points and the subsequent sequencing of the relative DNAs, it shows the loss of the Nokin DNA in the pathogenic variant and in the VUS, while the benign one was maintained. So as a conclusion, we have concluded to the fact that this strategy, it could be feasible for analyzing the trend of ATM variant persistence within cell populations, and more experiments are now ongoing in progress to improve our detection efficiency of Nokin and analyze single variants and cell fitness. So in the end, I want to thank all my collaborators and friends in the Unit of Cellular Networks and Molecular Therapeutic Targets at Regina Elena National Cancer Institute, Rome, Italy. And I want to say that in this project, we are 3. So I want to thank first our incredible boss, Dr. Silvia Saddu, and my talented partners, Giulia Federici and Dr. Giulia Calconi. And thank you all for your attention.
Nouha, thank you very much.
You're welcome.
I have still a lot of questions, but I need question here, from here. Yes, that right.
CRISPR is used in mammalian cell lines. It means the cell lines of breasts. We are using it in the dans le cadre du cancer du sein. De rien.
Any other question? Yes, there. Cyril and Shkur. Cyril, but rapid question, quick.
Okay. How can the CRISPR-Cas9 system be employed to study the interaction between the mammalian cancer cells and their microenvironments, if it's possible?
Yeah, it is actually used in this context to study the interaction between the breast cancer cells and, thank you. Within this context, we are trying to introduce specific variants and mutations in the mammalian breast cancer in order to study the cell behavior in the condition of radiosensitivity, chemotherapy, etc. So these are the first steps of the project, because it's not easy that you introduce a point mutation in the cell and you obtain homology or homologous repair. So now the first step is to set up an efficient strategy to obtain a knock-in. And once your mutation is there, you start to study the interaction of your cells with the microenvironment in any condition that you want to set up, in a condition of chemotherapy, radiotherapy, etc. So CRISPR-Cas9 is used to introduce the condition that you want to study the effect on breast cancer cells and its microenvironment. You're welcome.
Merci. Concernant la modification,
oui.
elle est devenue plus stable. C'est-à-dire, elle a été corrigée.
Non, elle n'était pas devenue plus stable en fait. On a introduit la mutation et on a vu que le variant d'intérêt, il est là. Mais après quelques jours, c'est-à-dire après quelques passages, la mutation elle a disparu. C'est-à-dire, elle n'était, oui, vers le wild type, oui. Après quelques passages, c'est-à-dire que il y avait quelque chose dans. Exactement, c'est ce que j'ai décrit par la variant specific cell fitness, c'est-à-dire la réponse, c'est la souplesse en fait de la cellule en vis-à-vis du variant qu'on a introduit.
Merci.
J'espère que j'ai.
Question rapide.
Oui. Oui. D'accord. C'était une méthode d'analyse qualitative pour voir si on a ou on n'a pas une modification. C'est-à-dire, qu'est-ce qu'on a fait? On a fait une analyse de restriction. On a cherché des enzymes de restriction qui ont un site, consensus site, dans la mutation d'intérêt. Donc, si la mutation d'intérêt, elle est là, tu ne vas pas voir ton fragment d'ADN qui est là, gel, qui تعمل le gel. عرفت نوريك التصويره نتاع الجيل باغ اكزومبل فيت في هنا باغ اكزومبل شوف انا الكورسور فوالا هنا فوالا سي لو وايلد تايب باهي دونك احنا اخترنا اون انزيم دو ريستريكسيون اللي ما عندهاش سيت في الفاريون دانتيري دونك يبدا فما لو وايلد تايب تبدا فما السيت كونسنسوس دو سيت انزيم اذا كان عندنا اللي هي تبدا ميوتي ما تقصش نورمالمون باش نبداو عندنا دو بوند في الوايلد تايب وعندنا اون بوند في لي كلون ايديتي مي كيما تشوف هكا السيريل كلها قدام تالي بارفيت كلها كوبي من الوايلد تايب دونك حاجه كاليتاتيف قبل ما نتعداو للسيكونسينغ والان جي اس جوست بور فيريفيي سي احنا في الفوا صحيحه ولا لا كيما تعرف ça coûte très cher le séquençage le NGS etc. donc on a développé une méthode qui est pratique rapide où le coût نتاعها اقل pour كيما l'analyse qualitative présence absence. De rien. Non. Because we, for example, another method to introduce a mutation. Because CRISPR-Cas9 has proved its efficiency to introduce point mutations. That's why we were using. But as you know, when you introduce a mutation, it's kind of stress. So when you have non-homologous unjoining, it's the frequent event. But when you have homologous repair, it's the special event.
I have then my question.
I know.
So
okay, we will talk later.
You have a stress, pyromycin.
Mm-hmm.
You found the perfect condition. So you.
No, pyromycin, it's just to check if our cells had integrated the plasmid or not.
So selection, voilà. For the selection, and then you remove the fact. You don't use anymore the pyromycin. And then at a certain moment, you then restart seeing the wild type phenotype. Okay?
Mm.
So did you check if when we use pyromycin again, if you have phenotype again ولا لا؟
ما جي با
قعدتوا ديما بالبيروميسين ولا جوست؟
احنا بدلنا كانت من الاول عملنا ترانسفيكسيون كي ستابل كي ان بروتوكول كي نو بعديك تعدينا للترانزيانت ترانسفاكشن معناها نتعديو ديريكتومون ابري معناها باساج في البيروميسين ديريكت نعملوا كوليكت دي سيلول اتسيتيرا معناها ما خليناش تقعد السيليكسيون على طول على خاطر هذاك اون كومبري اللي هو سي ان افي اللي خلينا نشوفوا الوايلد تايب اكثر من.
So to answer then Reeb question, you can use CRISPR or you can even use a transfection with a plasmid and to check. So it's kind of equilibrium between
both two.
But I do assume that all together we can have a discussion regarding that.
Then and we can answer your
Nouha,
question.
Thank you very much.